welcome you all in the class of computational biology and bioinformatics. So students, today we are going to do the practical on open reading frame prediction. Okay, we have done this practical before this also, so we are doing it again because some students have requested for that. Okay, so let's start the practical on open reading frame. All of you knows what is open reading frame? Just tell me, what is open reading frame? Open reading frame is the DNA sequence is open for the reading. So when the reading is start at the fixed starting point, means these are non-overlapping frame. So DNA sequence is read from fixed starting point, starting from the means trinucleotide, like 3 nucleotide makes 1 codon. So we start from position 1. Okay, we start reading that. So these open reading frame consists of non-overlapping codon. It should be read from a fixed starting point. Is it clear? Is it clear to you what is open reading frame? Just tell me. Give your response. All of you are here or not? Nobody is replying. Huh? Are you here or not?
Yes, ma'am.
So why are you not replying? Huh? I can't take practical. This is your practical class, okay? This is not the class. So you should reply. So you know the open reading frame, DNA sequence is transcribed into the mRNA. Okay? Then mRNA sequence contains the codon. So these for DNA also we define that codon for the direct strand and for reverse strand. So because there are the 3 nucleotide, means codon consists of 3 nucleotide. So we can start reading from first nucleotide, second nucleotide and third nucleotide. So these are called open reading frames, 3 open reading frames for direct strand. So same is true for the reverse strand. We can start reading from the first nucleotide, second nucleotide, third nucleotide. So each codon is consist of 3 nucleotide. Reading should be started from fixed starting point. Then you can say this is the reading frame, first reading frame, second reading frame and third reading frame. Now we will take a sequence. So we have the sequence, we have the file. We can upload it or we can paste here. This is the website of NCBI. In the Google I have typed ORF Finder. So ORF Finder is the name of tool which is used for the open reading frame prediction. Okay, so it will give you the multiple ORF, short ORF, long ORF. It will give you all the possible ORF. Okay, so you can consider the longest ORF as the significant open reading frame. Now you enter the sequence here, or there is an option, then we can choose. Okay, just from the download also we can take the sequence. This is our sequence. Already we have done practical on this. Are you able to see? This is the sequence.
Yes, ma'am.
Okay, so here you can see this is the sequence. Okay, so here you can see, you can start reading from the C or T or C. This is the trinucleotide. When we start reading from first nucleotide, this is called first reading frame. Then we start from second, it is called second reading frame. When we start reading from third nucleotide, it is called third reading frame. So, okay, because each codon consists of trinucleotide. This is the sequence from Homo sapiens So the name of the protein, the name of the gene is NF-κB repressing factor, NKRF. This is the name. Okay, and this is the variant one. Here it is written non-coding RNA or thing. It may be related to that. So we are taking this sequence. Let's see it contain the ORF or not. अब इसको कॉपी करते हैं. आपको दिखाई दे रहा है ना?
Yes, ma'am.
So I am pasting this sequence. I think इसमें कोई there should be no RF in this. Okay. So we will start. We are taking the AUG as a start codon. Okay. Now we are submitting. Okay. Let's see what happens here. So here you are able to see we got this result. Okay. So
Yes, ma'am.
okay. So this is the upper means on the upper panel you can see there is a figure and you can see the bar, color bar, which shows the length of ORF also. This is the sequence. Here you can see the nucleotide is written. See, you can see. Are you able to see? This is the range of the sequence. You can clearly see on the upper side. So this is the sequence, and on the sequence there are the different frames that are located. ORF24, ORF10. Okay, ORF10 is the I think longest ORF open reading frame. Okay, are you able to see?
Yes, ma'am.
So when you just click on this sequence or you set mouse on the sequence, you just get the detail annotation of the open reading frame. So this is the annotation of open reading frame. You can see this is the longest ORF. Shall I increase the size? Where it has gone, I think this one. So this is the longest ORF. ORF number 10 is the longest ORF, I think, because it is given. Okay, we will see also what कितने ORF है यहाँ पे. Location इसकी दी है 765 to 882. Okay, so this is the location of the gene and ORF on the gene or the protein that will be coded by this open reading frame that consists of 705 amino acid. Okay, so these are the number of nucleotides which code for the protein, and protein sequence you can see here. This is the these are the amino acid which is coded by this open reading frame. Are you able to see? Okay, I have already provided you the figure of this in the folder which is shared on the classroom. You can see the figure. So other ORF also you can see. These are the short ORF. If you click on this, if you set mouse on this, should come, I think. Why internet is not working? Yes, we have connection. We see. So you can take the figure of these also. Okay. Why it is not moving? Okay, so these all are the short ORF. These are clearly visible. ORF 24. So corresponding to the position, you can compare with the reference sequence. Okay, input sequence. So this is another ORF 17. You can see the annotation of ORF 17 also. So annotation of each open reading frame can be taken from here whenever you set mouse on the color bar. Okay, do you have any question regarding this? So you can download the ORF also by clicking here. Okay, you can download the FASTA sequence. So you can extract the open reading frame sequence from the gene sequence. Okay, but it is not the longest. So for the longest sequence you can show in your result. For the longest sequence you can download the FASTA sequence for the longest ORF by clicking on that. So here you can see we have obtained the file of longest ORF. Okay. We can open it in Notepad. Here, are you able to see? Are you able to see? The ORF 10, that is the longest ORF open reading frame, and we got the amino acid sequence of the longest ORF. Okay, amino acid sequence. So you have to do this. Students, are you able to see this? Are you getting? So this is very easy. So open reading 10, I will send you. I will upload it on the Google Classroom also. Okay. So these all are short ORF, open reading frame. You can also see the annotation of these. So you can get anything: coding sequence, location, protein length, position on master sequence, protein positions. So important is the means you can download the sequence of the open reading frame. Okay. So these are all the short reading frame, but the longest ORF is important. So you have to download at least ORF 10. Okay, you have to mention this. You have to describe this in your result. This is the longest ORF. So these annotation you can make the table for each ORFs. How you are going to make the table? See just here, this is the sequence for longest ORF. Amino acid sequence is given here. Okay. So here you can see. So in this way you can make the table also. Are you able to see? For the different open reading frame you can make the table like this: ORF 10. Strand, positive strand, frame number 3, start position and stop position. Okay. Students, are you able to see? So each open reading frame you can see here, and this is the annotation. Minimum annotation is shown here, and for the more annotation you can set the mouse on the color bar, means on that. If you want to get more detail, so you can add the detail from here. Okay. So you can add the column on this, and you can also make the different table. So make a different table. Just make one table from here. This is one table for all ORF. This you have to make as it is given here. So you have to make one table, one here, and you can also make the one elaborate table. You just make the one elaborate table also, in which you will you have to take the column from here, like in the ORF. We got so many details, so many annotation. For the annotation table you have to make the annotation table also. So that will be elaborate, that will consist of elaborate result. So first table format will be like this. This is the first table format, and figure you have to take here from here. Okay. Just you can okay, if you are not able to save the picture, कैसे आप save करेंगे? How you are going? You can do the print screen. Click on the print screen, like it is the key near to delete key. So you can do the print screen. You can paste on you can paste it on MS Paint. Okay, so in the MS Paint I am pasting this figure. So here, from here you can save whatever you want to save, you can save it here. I mean, it's like to save the picture. We can save it in some another folder for BBT 205 practicals. Okay, ORF. Okay, this is ORF figure. So we have saved. Or in your Word file you can import that figure. Okay, from the location you can insert that figure, you can crop that figure in the MS Word. You know how to crop the figure? So you can keep the figure as much you want. You can crop the size of the figure. Means whatever you want to show, just keep that portion. All of you knows? All of you know about this? Just reply me. You will do that. You insert that figure in your MS Word file. Then from there, from MS Office, you can crop that figure. Okay. So this is all about. Now from here you can also do the blast, smart blast or blast from here. So it will compare this sequence to the protein sequence that is stored in the UniProt KB database. So these are the different database. You can select any database for the comparison. So it will use the sequence alignment. So from here you can do the blast also. Now if you have any question regarding this, you can ask. So this was the practical of open reading frame. Okay. Do you have any question here?
No, ma'am.
So in the objective you have to write down means prediction of ORF. Then you explain the hypothesis. Then you keep the hypothesis, you just write down what is open reading frame. Just I defined you in the starting of the lecture, you just give the definition, hypothesis, what is ORF, how it is important, okay, what is the significance of ORF. Then just write down the methodology, how we have done the practical. You have to write down the method. Okay, or you can write down ORF finder, which method the ORF finder is using. Means you have to write down the steps how you are performing, that will come in the methodology. Step 1, 2, 3, how we have performed. We have retrieved the sequence. The first step is the retrieval of the sequence from the gene database. First you will retrieve the sequence from the gene database. Then you will paste that sequence into the window of ORF finder. Then you have to submit the sequence. All of you know how to retrieve the gene sequence from the NCBI. Okay. So do you have any question here regarding this practical?
No, ma'am.
Okay. Anybody from the class? So table you can make in the same manner as this data is written here. Na? These are the different ORF. You have to see it from here. You can download the different things. Just download the data from here. Just save the pictures. then use those things to write down your practical 5. You save this page also in the form of PDF. You can save this page, or you just save all the pages and before this step to input the sequence and for the next step also. So each step you have to save the page for the each step. Then you can write down your methodology. Okay. So we can go back, or we can do further. The blast analysis also we can do. But today we are discussing only open reading frame. Okay. So here some information is given here, like example have been given. So here you have seen we can take any choice of codon, whatever we want to take. We submit here. To write down the method, you can also read about the ORF finder, which program means how the ORF finder predict the open reading frame. So it is written here. You can read more on the ORF finder for the method section. So write down something about the ORF finder, which method it uses. Do you have any query you can ask? Do you have any query?
मैम जो पीछे वाली स्लाइड थी मैम, वो आपने मैम सेंड नहीं करी मैम पीडीएफ के फॉर्म में।
कौन सी?
मैम जो पढ़ाया था आपने पिछले लेक्चरों में मैम ऑनलाइन।
मिस अभी की बात यू आर टॉकिंग अबाउट नाउ? क्या मेंशन नहीं किया?
नो मैम जो पीछे मैम क्लासेस चली थी ना ऑनलाइन अभी,
हाँ
20-25 दिन, तो उसकी आपने मैम बोली थी पीडीएफ सेंड कर देंगे पूरी, सारे नोट्स एक साथ बना के।
मिस पीपीटी?
या मैम
आई हैव अपलोडेड दैट ऑन द ज्ञान संचय।
ओके थैंक यू मैम। मैम देखा नहीं मैम उसे हमने।
I have sent you the link also, miss sequence variation, gene therapy,
ओके मैम।
genome wide association studies that I have uploaded. I have uploaded the YouTube lecture also, and you will get the PDF also. So for the multiple video I have not given the PDF. So because that is saved in the one PDF, one or two PDF, okay? Because lectures are multiple for the same PDF, same PPT lecture, same PPT. I have taken the more than one lecture. So you just see on the Gyan Sanchay. Okay, you will get everything. So for the to add in the methodology section of method, you just read about open reading frame. What is the method of ORF finder? So you can search on the internet. Okay, for the documentation you can search on this website also, resources. Okay, there should be some more information about this, huh? What is the method of open reading frame? Battery is low now. Okay, it is. Method of ORF, we need to look method. So for the method you can find out this software is based on which method. So add something in the method section also. Procedure. Finding ORF, so different methods are given. You can look for, you can search on the NCBI website, you will get some detail how we find out the open reading frame. Method I have told you, we start from the first reading frame, that is the method. You can mention this, first nucleotide, second nucleotide, third nucleotide, the DNA sequence should be read from fixed starting point. So manually also you can find out the ORF, okay. I think it should be here on the website. So some detail is written here. Actually this notification is coming here, so I think it is. I mean behind this COVID notification you will find the detail of the ORF finder. Just search here. I mean how it will go. I think इसको हम कैसे skip करें, यहाँ से skip कर दें. So it is written something about the open reading frame. So for the program written the range of each ORF along with its protein translation. So you can write down the method of this ORF finder here. Okay, write it from this. इसको आप लिख देंगे, ठीक है. और इस पेज को भी आप सेव कर लेंगे. इसको भी आपको लेना है, मेंशन करना है यहाँ पे. यू जस्ट पेस्ट योर सीक्वेंस हियर, यहाँ पे, और तब उसको आपको आप यू कैन टेक मीन्स ओके नाउ आई डोंट हैव सीक्वेंस. तो आप इसको प्रिंट करके यहाँ से प्रिंट PDF करके आप सेव कर सकते हो उसे. प्रिंट, सो यू कैन सेव ईच पेज एज द PDF ऑल्सो, और यू कैन टेक द स्क्रीनशॉट ऑल्सो. Okay, so here in that way you will save, then use these saved pages, use the saved pages to write down the practicals. What happens? You just save whatever you do, you just paste your sequence from where we took the sequence. Okay, just copy this sequence. So you can show your method how you have done this practical. Okay, paste your sequence again, then save this page. Save the page so that you can use this for writing the practical file. Okay, just I'm telling you. So you can use these pages to write down the practicals. Okay, so from here you can write down about the open reading frame also, ORF finder, open reading frame finder. Okay, use this page to write down the methodology. Or next page, what will you find there? उसमें से आपको you have to write down the result. This page also you can save at the middle. Screenshot you can take whenever you will do the print screen, then you can paste the on the MS Paint and you will get that figure of the screenshot. Then you can save your figures. Okay, so use the MS Paint, then you can paste like this. If I do the print screen, I clicked on the print screen, or if I click on the here paint, now you can see. Are you able to see the screen here now? Now the screen is printed here. So you have to do the print screen, then paste it here, then save this figure. You can save this figure also. Whatever you want to save, so you. Okay. ORF sequence input. So we have given the sequence input. Now you tell me what is the open reading frame? Open reading frame, code for the probable sequence, that code for the protein. So it should consist of the nucleotide codon, trinucleotide codon. Okay. Trinucleotide make one codon. Each codon consists of trinucleotide. So there will be three reading frame for one sequence, one DNA strand. Maximum 3, because each codon is made up of 3 nucleotides. So there will be 3 possible reading frame for direct strand and 3 for the reverse strand. So there will be maximum 6 possible open reading frame. 3 for the direct strand and 3 for the reverse strand. Now you understood? If you have any more question, then you ask. Okay. Now this practical class is over. Next time we will do the BLAST practical. Okay. So nowadays we are revising the practicals to write down the files. You have to write down each big practical clearly in the given format that I have given you. Use the template that I have provided in the Google Classroom to write down each practical. Don't overwrite on the template. Save the original template, then save as that MS Word file in new, like practical 1, practical 2. Then write down the practicals. Save each practical. Or you can directly write down on the practical notebook in the hard copy if you do not have the computers and laptop at your home. But for some pages you need to take print, or you draw just figure. Like ORF figure, you can draw the figure also. This figure, if you can make it by your hand, then do it. Okay. So you can draw this figure. Will you draw this figure? Okay, so you can do this, or if you want to take the print, then you can take. So I think you can draw this figure also. You can draw and just show. On the blank page, white page, you can draw this figure. No need to take the print of this. Okay. For the protein visualization, for the protein structure, that also you can draw. The protein structure also you can draw, or you can take the print. I think it will look good if you take the